My name is Rion Naus. I'm a Stewardship Ambassador. I'm a certified public health educator. I'm a dad, and I own and operate We Grow With LLC. Really great sample, and I want to show you what's taking place here. We're going to go to the microscope. Ooh, there we go. And get that out of the way. You can see they're trapped. Oh no! They are poking and probing every little spot. Oh man, almost. Like, this is at 100 magnification. You can see all the other material has already been squished together as the slide has dried. And the larger creatures enjoying a little freedom of movement while it lasts. Oh, kind of looks like they're working together now. Oh, they did. Look at that. When we talk about aggregates, we don't often have an opportunity to see aggregate formation. But capillary action can do that with pressure, and it's called bioturbation. Does that with the living part. Now that edge there, it really is just what the surface tension of water is. And as that slide dries out, that tension, oh, look at it push. We're going to go to 400 and be able to see even better. You can see some amazing bacillus inside the sample. Those are the long thread bar shapes. You can see the fine hairs surrounding the body. Those fine hairs are called cilia. And because of that, this creature is a ciliate. Yes, it is. Any of those superfine hairs, like we have cilia in our own body, so this is a structural description. At this scale, form and function really does go hand in hand. You can see some of the other material making up the aggregates. Now, because we have the opportunity to look at the same scene but utilizing different observation tools, I'm going to go back out to 100 magnification. And then I'm going to utilize Darkfield. Darkfield is a way of having the light not just shine right up through the base, but actually come in from the side. And when that happens, things tend to glow. So you can actually see inside of those ciliates different things that they've consumed. Refracting the light differently. And if we go to 400, you can see how that aggregation is filled with bacteria and different minerals. When we talk about environmental impact, you can see how just this drying out process, which is natural, this drying out process drastically changes the microbiome. And so these creatures require the ability, and this is what like epigenetics is all about, to respond to their ecosystem, their changing ecosystem. Now, when the ecosystem itself, just like this water pocket here, when the ecosystem itself can't sustain life, then the organism tries to preserve life. And depending on the type of organism that it is, that preservation of life is a ton state if you're a tardigrade, or a cyst state. And it, in many ways, is like a suspended animation, because they're waiting for there to be habitat hospitality. They're waiting. They're waiting for the opportunity when they get to grow too. And at that point, like these small things that are taking place in a single drop mirror what we experience at our level on our scales. And so this protective state is when they say, all right, I'm not done yet, but I'm going to have to take care of myself for a little bit. Oh, so close. They think they might actually utilize that pocket, you know. We'll go back out just for a little perspective. That protective phase that they persist in, it initially seems counterproductive to an improved world, because it's still responding to the one that wasn't sustainable. So an example of that is when there's no more water, then they get rid of all the water, and then if there's no water left, they produce a waxy cuticle to protect the little bit of water that's actually required for them just to not die. And that waxy cuticle becomes hydrophobic. It's kind of an inside-outside phobic, which, you know, maintains the water it has, but doesn't want it. It's not going to get any more. Right? When we're dealing with houseplants, as an example, you'll go to water them after they dry out, and all the water just beads up and right over the side and onto the floor. And that's not because it's not thirsty, it's because everything that is thirsty protected themselves with a waxy cuticle, at which point you peed. You wet your plants. And so if you water just a tiny little bit, on the surface, damp, super, like not trying to water, but trying to wet, that will give the time necessary for that waxy cuticle to be metabolized and for the creatures to get back to work. When we talk about plants have a root and a root hair as their physical structure that allows them to reach out and probe into an ecosystem, into their ecosystem, whether it be the pot or the plains. And they'll fill that whole space, and you'll have roots that look like whatever you contain them in. This space, this size, the scale that we're looking at here, this is the mycorrhizal fungi, our arbusculars, the saprophytes, the creatures that are 10 to 100 times smaller than a root hair. This is the only water. This is the only water that exists in that system. It's all dried out. The mycorrhizal fungi can access even this interpore space fluid and be able to share that, service that to your plants. The nice part is if it's natural, then this system still continues to help make microaggregates, which are required for it's interesting. You can also see the viscosity, the thickness of the fluid itself having changed. So this is now, even though it's a single spot, there's a micro, wow, look at that, pressure pocket that is it's freaking cool. Like a diver's bell. So inside, this has a higher partial pressure. So this has a higher partial pressure of whatever gases are dissolved in it as well. You can see all of the different bacteria swirling around it as it continues to seek refuge. You can see the speed at which those little hairs churning around their whole bodies. So as that partial pressure increases, the different mineral components interact with different enzymes. So like all of these are cues of what's taking place and changing, and then signaling for the creature itself to morph, to change, so that it can actually survive this insurmountable moment. Usually I don't have the opportunity to record for this long. But this technology has allowed me to share more. Usually it requires like being here. Clips and snippets don't tell the story of watching this system, which is everywhere and fascinating. It's all real. It's all something you can look at, you can learn about. For some people it weirds them out because they've never thought, period, much less about it. And so instead of just turning this off and taking care of other things, it takes time to watch this process. But you can see the amount of energy that has been released into this system. Oh, look, there's some. What was that? Oh. Very cool. You can see what looks like other creatures large enough to be modal. Those I don't know. It actually might just be because of the compression in there that the vortices coming off all those hairs are drawing in. We're actually seeing it able to draw in from different directions. Or push off. Draw or draft. So in a few more minutes, when they can't move at all, we'll have the opportunity to see that transition. And then while we witness that, it, for myself, always creates a greater perspective on the rest of that aggregate we see closing in upon them. Because some of the things that, maybe initially thought were just a fulvic or humic acid, now we realize that fulvic and humic acid actually might have been a ciliate or a testate amoeba or some other thing in a protective state. I think Darkfield is pretty amazing for this. I think it creates a depth that Brightfield tends to wash out. Now, this small end of this larger aggregate, the whole slide is drying. And so it's not just the water. So there's a drawing of all those pieces together until some, they're too big. And so they cleave off and others are along for the ride and get squished together. I'm not sure if this one will get cleaved off or squished together because of how much separation those ciliates have caused. And so it might cleave off. And if you look at it, if you have a little raceway, that right there, when gentlemen like Martindale talks about capillary action, that pore space right there would be capillary action. And so if you don't have, and it's pore, P-O-U-R, that's why it's our space, pore space, And some people think about money and they go, oh, poor me. No, poor. P-U. P-O-U. So many things. O-U-R. So if it cleaves off, then we might see one of those possibly to kind of get wrapped up all by itself. Remember, it's resisting that surface tension where everything else is yielding to it. The amount of working that these little creatures have done, you get a sense of how well packed and man, anything not flattened and squished, looking for any give. So this video demonstrates many different facets and a few different microscope techniques, but really it's an observation tool. You need to be able to see. This is all what's really going on all over the place. When you water the lawn, everything is suspended and hydrated, and as it dries out, everything dries out, just like we're watching. But being able to observe allows us to serve in a much more relevant manner. It has a frantic feel to it. And I would assume that people would, anthropomorphize and say something like fear. But there's also part of me that just thinks that it's doing exactly what it would be doing if all the water was there. We just see it condensed. I think it's these other cues that causes the instinct to respond. Oh, let it go. You can do it. Come on. There's not over there. Keep going. Oh, it might be changing. Oh, do you see how everything got squished into the end of it? I mean, it might be that simple too, just as a pressure component, is that when it squeezes into a spot and everything gets squished into one part of it, and the pressure around it can maintain that, then it's a different, structural reaction or response. Whenever you hear people talking about biology, and they describe it and discuss it as if it's a technology, to the point that some people even just call it technology, it disconnects from this willing wiggle that, when we find ourselves tickled just right, we can enjoy too. Oh, that film layer is so thin that the other one can't get past it. So now we're talking about the thickness. You see how it bulbs out? Oh, Had to make room. I think that one's to a place of the inevitable. I think we have some cystate transition. So now we're at 400 magnification. You can still see some bacteria swirling around the ciliate. We want to watch the membrane exterior and the inside as it goes into that protective state. So these ciliates are part of a diverse group of single-celled organisms called protozoans. The hair, called cilia, the hair-like structures called cilia, which they use for movement, feeding, and sensing their environment. Found in almost any watery environment. They range from free-living species, like the paramecium. That is super cool. Usually you do not get such a sitting still opportunity to look inside of these creatures, which were once described as a race car. But you can see that inner structure